Okay, Assalamu alaikum everyone. Good afternoon, Jum'ah Mubarak. Hope everyone's doing well. So I believe I see a lot of familiar names. So nice to see you guys again. For those of you who don't know me, my name is Raffi. I'm a final year medical student. And if you were here in the pre-med program over the summer, I taught the exact same lecture. And here there's just a few small little details, but if you remember anything from that lecture, you're pretty much set. So this lecture is about protein sorting and trafficking, and we'll get into what that means. But yeah, so I know most people don't usually like to unmute to ask questions. Personally, I prefer if you do, but if you want to ask any questions at any point, just do so in the chat, and I'll try to answer when we go through the checkpoints. So here are the objectives. So based on the doctor's lecture, uh, there are all these objectives, and honestly, 7 objectives seems like a lot for a 20-slide PowerPoint. But we'll just keep it simple. We're just going to talk about what does it mean when we traffic a protein, where the target sites that we send them to, like lysosomes, nucleus, mitochondria, peroxisomes, glycosylation, what that means, the difference between constitutive and regulated secretion, and then some clinical correlates. Now, a fun fact about this lecture is when I was in first year, around the same time as you guys back then, I did not like this lecture at all. Matter of fact, it was one of those lectures I ignored. Um, but I like lectures like this for PAL because it forces me to actually understand what is actually really easy. So I hope, Yani, by the end you'll realize how simple this is. Okay, the truth is this isn't really a biochemistry lecture. This is a physiology lecture. And physiology, I'm sure if you, Yani, it's been 8 weeks, 9 weeks in. I'm sure you now understand what it means when we say the word physiology. And if you don't somehow, it's the study of function. And the study of something functioning is we're talking about the normal function. This function relates to disease. So essentially, we're studying what is going on. And if that doesn't work, what is the disease that happens? Now, we really need to focus on just function because again, you guys are still early in medical school, so you only need to understand what the function is. Later down the line, you learn the dysfunction, and we'll only be talking about dysfunction in a superficial sense. So if you can remember the process, you can remember what goes wrong if you know the steps. Okay, now, fun fact about me is I love Legos. I actually have a bunch of them on my desk. This is the latest set I got. It's an R2D2. Love it. Um, but I really love Legos, and I thought let's unpack this like we're unpacking a Lego, and let's make this fun. So we're going to talk about what is protein trafficking and sorting, how do we incorporate a ribosome into the rough endoplasmic reticulum, and we'll talk about what that means, the sites of protein trafficking and sorting after synthesis, and then other details. Okay, so what does it mean when we say we want to traffic a protein? Basically, let's talk about the central dogma of molecular biology. We make DNA, DNA makes itself. DNA will get converted to RNA by transcription, and RNA will get converted to proteins by a process called translation. Now, translation happens on ribosomes, and ribosomes, they can either be free within the cytoplasm or part of the rough endoplasmic reticulum. So ribosomes, which do translation, are either free in the cytoplasm or they're found in the rough endoplasmic reticulum. Now, they can go to different parts of the cells. So, for example, they can go to the Golgi, they can get modified, they can go into vesicles. And we talk about the difference between vesicle-mediated and free transports and a little bit of details on those. So we just need to talk about the triggers. What kind of forms does it take? And then some destinations that it goes to. So do you need to memorize this, Yani? I want Yani guesses in the chat. Do you think you need to memorize this right here? Yeah, no. Okay, good. Yeah, you don't need to memorize this. So another thing that you guys should do whenever you're approaching any sort of lecture and you're having a difficult time with it is identify what is likely to be asked about, what's likely to not be asked about. And realistically, I don't see how they could ask you about this. Like in an SAQ, for example, write the import sequence and you just get some random guy typing met there. It doesn't make sense. So this is just for the sake of completion that we have signals, and the signals, they come down to the amino acid sequence and they tell us where something's supposed to go. But I don't want you to memorize specific triggers. Please play it. Now, why do proteins go to the endoplasmic reticulum or the ER? Because they're sick. Anyways, manina. But we're not talking about proteins going to the endoplasmic reticulum. We're talking about proteins going through the endoplasmic reticulum. So essentially, all proteins, they start translation on free ribosomes. Like, so every ribosome initially is free. So when we mean free, doesn't mean it means no, it's in the cytoplasm. So we have all of these ribosomes in the cytoplasm, and when a mRNA sequence leaves the nucleus to go to the ribosome, it starts getting translated there. Like it. Now, the ribosomes themselves, they may remain free in the site. They may stay in the cytoplasm, or they can go to the rough endoplasmic reticulum. Or as a small side, fun fact, it's called rough endoplasmic reticulum because the ribosome go and attach to it. That's what makes the rough surface of it. Anyways, so if it goes to the rough endoplasmic reticulum, that means it's vesicle mediated. So when something goes to the rough endoplasmic reticulum, it will be transported in the form of vesicles. And vesicles are like these small carrier membranes, honestly. It's just very small carrier membranes, and they can be used for transport. So if something goes through the rough endoplasmic reticulum, it therefore must go to the Golgi, and that's before it reaches its target. And some of the targets, if we're using this, would be the membrane, outside, lysosome, back to the endoplasmic reticulum, and it uses SRPs. Okay, let's simplify this a little bit more. So we said in the beginning, ribosomes, they're either in the cytoplasm or they can bind to the rough endoplasmic reticulum. So if it binds to the rough endoplasmic reticulum, it will do something called vesicle-mediated transport. Vesicle-mediated transport means it uses these vesicles, and these vesicles are made by the rough endoplasmic reticulum. And it will use those vesicles to go first to the Golgi. And if you remember, the function of the Golgi is to modify proteins. So it modifies proteins that come from the rough endoplasmic reticulum through vesicles. And therefore, it can go to specific sites, which include the membrane. It can leave the cell. It can go to lysosomes or back to the endoplasmic reticulum. Each one has its own signal. And in order for the rough endoplasmic reticulum to get a ribosome attached to it, it uses something called an SRP. SRP stands for signal recognition peptide. So if a protein has a signal recognition peptide and it's being translated, or if mRNA has a signal recognition peptide and it goes to the ribosome to be translated, that ribosome will bind to the rough endoplasmic reticulum. And I have pictures of how this looks like at the end, because I think maybe this point is a little bit confusing. Now, let's say a protein doesn't have a signal recognition peptide. Will it go to the rough endoplasmic reticulum? The answer is no. It will remain in the cytosol, and it will not use vesicle-mediated transport. So it can go to sites like the mitochondria, the peroxisomes, the nucleus, or just remain in the cytosol. So let me just break this down again, just so we're all clear. mRNA goes to a ribosome. The ribosome itself will translate the protein. This ribosome can go to the rough endoplasmic reticulum if the mRNA sequence has an SRP. And if it goes to the rough endoplasmic reticulum, it will use vesicles to move around. It will go to the Golgi, get modified, and it will go to other sites, which include the membrane, outside the cell, lysosomes, or back. If there is no signal recognition peptide, the ribosome will remain free in the cytoplasm, and the proteins that are made will either go to the mitochondria, the peroxisomes, the nucleus, or just remain in the cytoplasm. So I hope that makes sense. Any questions so far? Is everything clear? All right. Perfect. Okay. Let's move on to how do we get a ribosome into the endoplasmic reticulum. Okay. I want you to ignore these numbers because these numbers and these words, they're very confusing. And I just want you to focus on the picture. And as you can see, this is the handwriting that made me decide that medical school is right for me, for the record. This is actually some of the best handwriting I've ever had. Anyways, so we have this mRNA. Hello, this mRNA is being translated on this ribosome right here. Now, subhanAllah, this mRNA expresses something called a signal sequence, or an SR, expresses a signal sequence which binds an SRP. So if it expresses a signal sequence, it will go to the SRP. And therefore, because we said the word SRP, we know that this is going to be vesicle mediated, or it's going to bind to the ribosomes, it's going to go to the Golgi, and everything we talked about in the previous slide. So this is how we're tying things together. So what's going to happen is this mRNA is going to be translated on this ribosome, and it expresses a signal sequence. The signal sequence is then going to attract an SRP. The SRP is going to bring it to this channel over here, that's translocon, and it's going to bind it so that the mRNA, rather than going outside, it will go inside here. So the goal is, let's make the rest of the mRNA, or the protein that's being translated, go inside the rough endoplasmic reticulum. So it does this, and then so you see it's a GTP bound, so it has to break the GTP to open the translocon. And if it's breaking GTP, think of it as just another form of ATP. I mean, it's not what it is, but just in the sake that it's an energy currency. This means that this channel is energy, it needs energy. Like I'll repeat everything again. So it removes the signal gets removed, and the mRNA is being translated to proteins into here. Okay, so let's restart. This is the mRNA. This is the protein it's translating. So this protein, when you translate this mRNA, it makes a signal sequence which attracts the SRP. Now, because it's an SRP, this means that this ribosome will become bound to the rough endoplasmic reticulum. Anyways, so this SRP is going to attract the ribosome to the rough endoplasmic reticulum, and we need to open this channel so that the protein that we're making can go inside the rough endoplasmic reticulum. How do we open this channel? We hydrolyze the GTP into GDP, and this makes energy, causing the channel to open. And we essentially synthesize the protein inside here. We remove the signal, of course, the signal sequence protein that we made. And this is pretty much the process of how a ribosome becomes bound. So let's look at the words. So this is just to keep it simple. First thing is we make mRNA is translated to the signal sequence, which we can see over here. The SRP is going to bind the signal sequence, as you guys can see here. The translocon needs to be opened. So now this ribosome is here. We need to open this translocon. So we break the GTP. This causes energy, which opens the translocon. We remove the signal. The protein gets translated inside. And now, where will this protein go? It will go through vesicles to the Golgi, and then whatever sites it needs to go beyond. I know there's a lot of words. Do you guys want me to repeat anything, or does this seem clear? Now, I want you to focus on the monkey explanation, because if a monkey can understand it, we all can understand it, I hope. All right. Can I re-explain? Which part would you like me to re-explain? This right here will previous slide. Okay. Wait. Okay. Let me show you guys how the previous. So someone asked me to re-explain the previous slide. So let me show you how the previous slide can tie into this slide. So we'll just go through them one more time. Okay. So essentially, proteins will start translation on ribosomes. So the mRNA, as you guys can see here, goes on a ribosome to get translated into protein. Now, if the protein expresses something called an SRP, this SRP will cause the ribosome to bind to the rough endoplasmic reticulum. Now, if it binds to the rough endoplasmic reticulum, it just means that the protein will be synthesized inside of it. It will use vesicles to go to its targets, which include the Golgi, and then the Golgi will modify it and send it out also by vesicle to either the membrane, outside, the lysosome, or back. If it doesn't have the SRP, it doesn't do this. The protein just remains in the cytosol, and it can go to whatever sites it needs to go, in a nutshell. Now, let's say it does express an SRP. This is what happens. The mRNA gets translated. It makes a signal sequence. The SRP binds the signal sequence, and brings the ribosome to the rough endoplasmic reticulum. The ribosome now needs this channel to be open. So we break this GTP, causing energy to open this channel. This channel will then the protein will then be translated inside. So the protein that we're making, it's going inside the rough endoplasmic reticulum. And we have this unnecessary signal sequence, so we just cleave that. And now the protein is being made, and now the ribosome is rough endoplasmic reticulum bound. So this is it in a nutshell. Is everything more clear now? All right, perfect. Another thing, now this is a question for you guys. Let's say this isn't a signal sequence. Let's say there is no SRP and there is no signal sequence. In your opinion, what do you think will happen? And feel free to unmute your mic or chat, whatever you prefer. So there is no signal sequence and there's no SRP. What do you think will happen? Okay, so Maha says it will go to the cytoplasm. Do you guys agree? Yes, it will. Yeah. So all of you guys follow up. Slave said it will be synthesized on free, stay in cytosol. Exactly. Good. So you guys understand this point. And no SRP does not go to the ribosome, stays in the cytoplasm. MashaAllah. And you guys are smart. You don't need this PowerPoint. So, yalla. مشينا. طيب, targets of protein synthesis. This is the hardest part of the lecture. الحين we go into the specific sites. But if you guys understood these two, the rest of it is somewhat smooth sailing. طيب. But this is the somewhat part. Now, lysosomes. Now, I want you guys to guess. Lysosomes. Is this going to come from a protein that's that's made in the rough endoplasmic reticulum or free cytoplasm, free cytoplasmic ribosome? What do you guys think? So if a protein is going to go to the lysosome, is it going to come from the rough endoplasmic reticulum or is it going to come from, yeah, okay, like you guys said it, endoplasmic reticulum, ahsant. Good job. So yeah, this is coming from the rough endoplasmic reticulum. This means that the protein making it has an SRP. This means that it's using vesicles to transport. Right. And this means it goes to the Golgi. So the Golgi, the basic function of it is to modify proteins. And this is what it's doing to the proteins here. So the protein is getting sent to the Golgi. In the Golgi, it binds mannose 6-phosphate, M6P. So it's going to bind it. And then for some reason that we don't actually understand, it just gets to the lysosome. That's literally it. The protein binds mannose 6-phosphate. And because it binds mannose 6-phosphate, it can then therefore be accepted by the lysosome. Now, one thing to note about mannose 6-phosphate that might be important is mannose 6-phosphate comes from, if you couldn't tell, mannose, and mannose has to be phosphorylated. So we need to add a phosphate to it. And this is done by N-acetyl glucosamine 1-PT. I'm sure there's a shorter name. Yeah. Okay, how do we know that the protein came from the endoplasmic reticulum? Someone asked this, and that's a good question. This right here. It's just that if it's going to the membrane, or it's leaving the cell, or it's going to the lysosome, or it's going to the rough endoplasmic reticulum, this means it came from the rough endoplasmic reticulum. If it's staying in the cytoplasm, or going to the mitochondria, or going to the peroxisomes, or going to the nucleus, that means it's coming from a cytoplasmic. So this is just something you need to memorize. But good question. Okay, so we make mannose 6-phosphate by N-acetyl glucosamine 1-PT. And I'm sure there's a shorter name for this, but حاليا I don't know it. And yeah, so mannose 6-phosphate binds the protein in the Golgi. And for some reason, the lysosome decides, yeah, you know what? Mannose 6-phosphate, give me 14 of them. Okay. Now, how do they get to the nucleus? The nucleus needs protein. So if it's going to the nucleus, does it come from the rough endoplasmic reticulum or does it come from the cytoplasmic ribosomes? Is mannose as I didn't know. Okay. So just yeah, it does come from the cytoplasm, but someone asked the question, is mannose added in the rough endoplasmic reticulum and phosphates added in the Golgi? No. So let me go back really quickly. The protein itself comes from the rough endoplasmic reticulum. It gets sent to the Golgi, and at the Golgi, mannose 6-phosphate, this compound, is going to be added to the protein. The only thing I mentioned is that we add a phosphate to mannose to make mannose 6-phosphate, and this also happens in the Golgi by N-acetyl glucosamine 1-PT. But for the sake of how does it get accepted by the lysosome? The protein binds mannose 6-phosphate in the Golgi and then goes to the lysosome. So I hope that's more clear. And you guys are correct about this. So if a protein is going to the nucleus, it's coming from the cytoplasmic ribosomes. So how does this happen? So importin and NIR, nuclear import protein, are the same thing. So NIR, nuclear imports protein, importin, same name, same thing. They're talking about the same thing, which is this S-shaped thing right here. So what happens? The protein that we made, the beautiful protein which came from the cytoplasm, is going to bind NIR, or importin, or nuclear imports receptor. And then it will go across the nuclear pore. So if you guys know the structure of a nucleus, it has pores, and it has these holes, and proteins can go through these holes. So that's what's happening here. So the protein binds importin, the importin takes it across, and it binds something called RAN-GTP. So it binds RAN-GTP. RAN-GTP is going to bind this S-shaped thing, which is importin over here. When RAN-GTP binds importin, this pushes the protein out into the nucleus. So in Hain, we've delivered the protein into the nucleus because we pushed it out by RAN-GTP. So let's just recap these first three steps. The protein will bind importin. Importin will take it across the nuclear pore. And RAN-GTP will then bind the importin to push the protein out into the nucleus. So hello, at this point, we've delivered the protein. What happens now? How do we take importin back out? The RAN-GTP is going to leave the same way it came in, through the pore, and then they're going to separate. So the RAN-GTP and the nuclear import receptor will separate. So let's recap all 5 steps. Step 1, protein binds. Step 2, importin moves it across the pore. Step 3, RAN-GTP binds importin, causing the protein to be released into the nucleus. Step 4, that leaves the pore. Step 5, they separate. Play. What about the mitochondria? So the mitochondria is a little bit weird. So we have a few players here. We have Tim, we have Tom, we have HSP70, and we have all these things. And as you guys may or may not remember or know about the mitochondria is that it has 2 membranes. It has an outer membrane and an inner membrane, and it has a space between them. So TOM is a translocon of the outer membrane, and TIM is a translocon of the inner membrane. So OM, outer membrane, IM, inner membrane, makes it easier to remember. Let me put this over here. Yeah. Okay. So what's going on here? First of all, the protein needs to bind something called HSP70C. Now, anyone can tell me what does the C stand for? Can you guys guess what the C stands for? Cytoplasm. Very good. So the C stands for cytoplasm. So what's going to happen is that this protein will bind HSP70C on the cytoplasm, and this will cause it to enter TOM. Now, what happens is that for some reason, after you bind HSP70C, it just causes ATP hydrolysis. So this causes the protein to be released by HSP70, and it allows it to enter TOM properly. Okay, how does this work? Think of HSP70 as it's also called a chaperone protein. And if you guys know, a chaperone is the kind of person that holds your hand when you're a little kid, making sure that no strangers talk to you and no one tries to like kidnap you or anything like that. So this is what a chaperone does, and HSP70 is a chaperone protein. So it's going to hold the hand of the protein. It's going to take it to the TOM. And the thing is, HSP70 can't enter the TOM. So what happens is that it will cause ATP hydrolysis to separate the protein from HSP70, and this drives it into TOM. And I'll show you what it's kind of like a snake-like movement. Now, it will then enter TIM, but now we need to get the entire protein into the mitochondria. So HSP70 will hold its hand again, HSP70M, which means mitochondria, and it's going to cause ATP hydrolysis, pulling it inside. Now, on the exam, they're not going to ask you who's holding whose hand and all that, but it looks something like this right here. Okay, so the green highlights where the HSP70s are. We have HSP70C and HSP70M. And the protein, in order to enter HSP70C, it needs, in order to enter TOM, it needs to break away from HSP70C. So this is one point of ATP hydrolysis. Now it enters here, and it needs to get pulled inside the mitochondria. And it does so by breaking HSP70M again at this point. Now, I know this can be a little bit confusing, but there's going to be an easy way to remember all this at the very end. So I'll need you to stay tuned. So what's just to summarize this, what's going on? HSP70C, the protein will bind HSP70C. HSP70C will provide energy for it to enter TOM. It then enters TIM, now needs to get pulled again by HSP70M, ATP hydrolysis. Now, I don't want you guys to remember this as it. I will give you guys a much easier way to remember it because it's very confusing. But until then, can you guys remember the words? So is HSP70 is in the inner mitochondrial membrane? No, it's in the mitochondrial matrix. Good question. So what I want you to remember is that Tom has an HSP70, Tim has an HSP70. The HSP70s will have ATP hydrolysis, and this is what causes the protein itself to move. If you can remember these three sentences, you are set for the exam. So can you guys remember those three sentences? Yes, alhamdulillah. Okay, great. Can you repeat the sentences, please? Of course. So Tom and Tim have an HSP70. The HSP70 provides ATP hydrolysis. This allows the protein to move. Simple or not? Is that simple enough to remember? Perfect. Okay. Now, last site we need to talk about are how do proteins get into the peroxisome? Um, okay, peroxisome is very simple. We have a protein called PEX5. So peroxisome 5. Play it. So cargo, by the way, they treating this like it's a warship. It's not really a warship, to be honest. It's a protein. They're making it kind of deep for some reason. I don't know why. Anyways, the cargo refers to the protein itself. So the protein binds PEX5. And PEX5 is going to enter the membrane of the peroxisome. What happens here is you have this sequence you have this sequence over here, this N-terminal sequence, and you have this thing called ubiquitin ligase. So ubiquitin ligase is going to separate PEX5 from the cargo or the protein, and it remains in the cytoplasm. And then we take it out through ATPase. So that's pretty much it. Just to repeat, protein binds PEX5. PEX5 enters cytoplasm, it ends the peroxisome. PEX5 and the protein separate by ubiquitin ligase. PEX5 leaves by ATPase. And it's written over here. Does that make sense? Yes. Yes. Great. I'll get to the two slides before this, the nucleus and the lysosome very shortly. Step 2. Okay. Step 1, the protein binds the PEX5. Step 2, the protein enters the peroxisome. Step 3, they separate. Step 4, the PEX5 leaves. That's it. In a nutshell. Okay. So in your slides, it mentions PEX2 instead of PEX5. Is this a typo? No, there's actually, I think there's around maybe, there's hundreds of different PEX proteins. So PEX2, PEX5, not too big of a deal. PEX2 is relevant for one disease where it's more affected than PEX5, but that's a little bit deep. But if it'll make you happy, just remember PEX. Not even PEX5, PEX2, just PEX. PEX5, PEX2, they both do the same thing. But PEX2 is affected in different diseases than PEX5. For the deeper understanding of it. But very good question. Thank you for bringing it up. Play it. So here we have a question, and I'd like someone to give me what they think the answer is. So I'll give you guys a minute to try come up with the answer. All right, I see someone in the chats. So someone says D. Okay, can you back up your answer? So if you're gonna say D, why is it D? No vesicles. Absolutely. Very good. No vesicles. So if, for example, um, they said they cannot use vesicles. Like the nucleus, proteins that go to the nucleus don't need vesicles. Proteins that go to the peroxisome don't need vesicles. Proteins that go to the mitochondria don't need vesicles. Proteins that go to lysosomes do need vesicles. So if a cell cannot make vesicles, it therefore cannot go to the lysosome. Very good. And now this takes me back to the point where I wanted to re-explain lysosomes. So I was asked to re-explain this slide and this slide over here. So with lysosomes, all that happens is that in the Golgi, protein binds M6P, mannose phosphate, and that causes the lysosome to take it. That's it. That's all that happens. It's being transported by vesicles, and then some details about how we made M6P. That's it. How do proteins get to the nucleus was another point that I was asked to re-explain. Protein binds NIR. NIR moves across the nuclear membrane. It gets RAN-GTP will bind to this complex, releasing the protein into the nucleus. RAN-GTP and NIR will leave, and they will separate. That's all that happens. So there was someone who was asking me to explain this. Does that make more sense now? Perfect. Okay, let's get to the second question right here. So give me your answer and then back it up as well. Because I know it's very easy to just like yell an answer, but I want you to be able to tell me why. Mannose phosphate is added in the Golgi. Okay, so you guys ignored my instructions about explaining why. So tell me why. Oh yeah, I get it. You guys are all very smart, mashallah. This I want to know why. I want you to prove your intelligence, or at least like make sure that you guys understood the lecture. That's why I'm here. I know chat's cooking right now, so because yep, very good. So M6P helps the protein enter the lysosome. That's it. It really comes down to this. Now, I want to ask you guys something. At any point in these two questions, did I ask you to recite to me the steps of something? No, I didn't. Do you think on the exam they will? They could, but most likely not. So the key to doing well on very difficult lectures is something called buzzwords. And I'm sure you've heard of the idea of buzzwords before, but you want to remember what words go together. So for example, if I say words like N-acylglucosamine, mannose-6-phosphate, vesicles, I'm talking about the lysosome. If I say words like RAN, important, NIR, I'm talking about the nucleus. If I'm saying words like ATP, TOM, TIM, HSP, I'm talking about mitochondria. If I'm talking about ATP, PEX5, PEX2, as one of your colleagues mentioned, ubiquitin ligase, ATPase, I'm talking about the peroxisomes. If you remember this slide right here, it pretty much will explain all of these slides over here. And that's the thing. You need to identify. This is something I want you guys to practice on your own. And when you see a lecture and you're having a difficult time understanding, don't just cram the steps and try to and memorize them. There are points in your life where you will need to memorize steps for something, but this isn't going to be one of them. This is biochemistry. Everything is a process. Everything has a million steps to it. If you're going to remember every process, you're not going to remember it well. Now, someone asked me to explain the previous question. I'm more than happy to do that. So over here, we said that basically these cells can use vesicles to transport proteins. And which of the following items below is related to this? So what we're asking for here is which of these is related to something that uses vesicle-mediated transport. Now, in terms of M6P, NIR, HSP70, TOM, TIM, PEX, I'm talking about sites of where the protein is supposed to reach. So I'm talking about the lysosomes or the nucleus or the peroxisome or anything like that. So from these sites, which one of them uses vesicles? And the answer would be lysosomes. And lysosomes, I remember the words like N-acetylglucosamine, vesicles, and mannose-6-phosphate. And here, mannose-6-phosphate is here. So it's simply because it uses vesicles, that means we're talking about lysosomes that can be reached, and lysosomes use mannose-6-phosphates on the proteins to allow them to enter. Does that make more sense? If you want, I'm more than happy to repeat. I can try to simplify it, but hope that makes sense. Okay, perfect. You got it. Of course you did, because you are very intelligent. Okay, we have some other information. This rest of the stuff is really very simple. We have something called glycosylation. Now, the word glyco should tell you that we're talking about sugar. And by sugar, referring to glucose. So pretty much, glycosylation is a form of post-translational modification. The word translation, that means how do we make proteins? And then post-translational, so we modify this protein after we made it. And we do this called glycosylation, which adds glucose to proteins. So we have something called N-linked and O-linked. N-linked uses arginine and the endoplasmic reticulum. And O-linked is on serine or threonine in the Golgi. So just remember, if we're talking about N-linked, this is happening on arginine. This is happening within the endoplasmic reticulum. If we're talking about O-linked, this is happening on serine and threonine, and it's happening within the Golgi. So O, 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 N, N, N. Try to make that connection. Now, post-translational modification is important because it allows proteins to fold, and proteins folding is important for their function as well as their structure. And this happens for collagen. Other forms of modification that happen with collagen include hydroxylation, proteolytic cleavage, and cross-linking, which we don't need to know for this lecture. You have constitutive and regulated secretion. Constitutive basically means it's always happening. So this protein's always being made 24/7. An example of this is collagen. Regulatory means that we're making this as needed, such as insulin. And then here is a summary. So I like doing these animations. And because you guys are very kind, you're going to tell me that this is the best animation in the world and Pixar should hire me. But we'll ignore that for now. So what's happening? We made an mRNA, and the mRNA is going to go to a ribosome. And we're just going to use two ribosomes as an example. Now, this ribosome is making a protein over here, and it doesn't have an SRP, so it'll stay. But this ribosome has an SRP. So you guys will know what happens later. Now, because it doesn't have this protein, this ribosome isn't making an SRP, that means that the protein will stay within the cytoplasm. If it stays within the cytoplasm, that means it could either go to the mitochondria or the peroxisome or the nucleus, or it can just remain free within the cytoplasm. But if it does have an SRP, it's going to go to the rough endoplasmic reticulum, and it will transport by vesicles to the Golgi, where it will get modified and go to the lysosomes, also go to the membrane, and leave the cell, or go back to the rough endoplasmic reticulum. And this summarizes everything we talked about. Now, some clinical correlates that you guys need to know. So we have eye cell disease, mucopolysaccharidosis 2, basically a deficiency of N-acetylglucosamine. This means it cannot make mannose-6-phosphates. Zellweger is a mutation of PEX, specifically PEX2 and PEX5. But it affects peroxisomes, causing them to be empty. Menkes is an issue with lysyl oxidase. It affects cross-linking, which is a form of post-translational modification. And we talked about another form of post-translational modification, which is glycosylation, and that affects collagen. And then we have cystic fibrosis, where we get a bunch of misfolded proteins that are not COTS. Now, this is our lecture. We're done. We have a question, and I'm more than happy to explain anything that doesn't understand until we get to the question. So can you repeat the animation? Sure, I will get to that. Can you explain the slide Slim with Tim, Tom, animates more? Okay. I don't want you to know, I'm just going to say it like here. I don't want you to know Tom, Tim, how like HSP70 holds its hand, breaks up, all that. No. I want you to remember a few things. Right? First of all, you have Tom and Tim. Tom is on the outer membrane and Tim is on the inner membrane. Right? And near each Tom and Tim is an HSP70. The protein, when it gets to Tom, HSP70 will cause ATP hydrolysis, allowing the protein to enter Tom. And then when we get the protein will reach Tim, HSP70 will cause ATP hydrolysis to allow the protein to keep going. But I don't want you to remember all the hand-holding stuff because that's kind of weird. But I want you guys to remember the three sentences I talked about. HSP70 is found next to Tom and Tim. HSP70 causes ATP hydrolysis. This allows the protein to move. That's it. And this is where HSP70C is, HSP70M is. This is Tom, this is Tim. This is all you really need to know. You don't need to know like the crude hard details of this. So do you think you understand this a little bit better? Okay, great. And then we will repeat the animation. Sure. Okay. So does anyone want to be brave enough and unmute the mic and help me out here? ولا chat ولا anything? All right. So that's fine. We'll um we'll go with me explaining. So basically, we have a protein over here, and the protein is going to go to the ribosome. The ribosome is going to basically have this mRNA, and it's going to be translated over here, as we can see. And basically, we're talking about two ribosomes over here. We have this ribosome, which does not have an SRP, and this one which does have an SRP. So the one without an SRP is going to go to the mitochondria, or it's going to go to the peroxisome because it will remain in the cytoplasm, or it will go to the nucleus, or it will remain in the cytoplasm. Then we also have this one, which does have an SRP, and as you guys remember, it will go into the rough endoplasmic reticulum. And when it goes here, it's going to transport via vesicles to the Golgi, in which it can go to sites such as the lysosome, the membrane, outside, and then back into the rough endoplasmic reticulum. These are the four sites it can go to. So I hope that's more clear now. Any questions? Perfect. We have this last question. So I'll give you guys a minute to read it. And let me know when you guys want to see the options. Can you show the options? Right. I'll wait for a few more people to say that they want the options before I move to the options. But just take a screenshot of this, or like take a picture of this, and then we'll try to show you the options. So we'll give you guys a minute to do that. So just take, all right, 3, 2, 1. Here are the options. Okay. If you're going to say an answer, I'd like you to justify it. So here's the question back. Here's the options again. All right. Another person said an option and justified it. Thank you. All right. So the answer to this is C, synthesis of cell membrane proteins. Now, why is it C? Why is it not the other options? So here, essentially, we're saying, Annie, the thing is, sometimes in questions, what they're going to do is they're going to give you this long paragraph, which is useless. You just need the last two sentences. Cells that are exposed to longer ischemic periods are found to have reduced numbers of ribosomes attached to the endoplasmic reticulum. So we have less ribosomes bound to the endoplasmic reticulum. This means we're not making proteins that would go to sites like the lysosome, the cell membrane, these leaving the cell or back to the rough endoplasmic reticulum. So we're looking for anything along those lines right here, which is cell membrane proteins. ATP production, drugs detoxification, cytosolic proteins, steroid hormones, cytosol. I mean, this all happens with ones that are not bound. And you're probably wondering, okay, I didn't talk to you guys about drug detoxification. and I didn't talk to you about steroid hormones. And you're right, I didn't. However, you also need to be aware that to answer what you do know, there is going to be an option that you do know or you should know, and there may be some other distracting options just to throw you off. And that's the thing. Sometimes questions are difficult. This was a very difficult question, actually. And I got this from something called U World, which you guys will learn about for any 3 years from now. But yeah, if you're able to answer this, you should be ready for you're more not just ready for your exam, you're ready for other exams too. So yeah, that's it from my end. I think I yapped enough for you guys. And you have a question regarding E. Okay. Synthesis of steroid hormones, smooth endoplasmic reticulum. That it's done by the smooth endoplasmic reticulum. It's just there to distract you. Same thing with drug detoxification. So that's just something to keep in mind. These are both done by the smooth endoplasmic reticulum, and they're just there to distract you. Okay. I think you meant cytosolic proteins, but
Yes, I
let's
did.
okay, no worries. So cytosolic proteins, they will either stay in the cytoplasm, or they will go to the nucleus, or they will go to the mitochondria, or they will go to the peroxisome. These four sites. Like, let's
Oh,
make
okay,
this,
thank you.
of course, let's make this easy. You need, I would say, if you like are really short on time, this slide, this slide, this slide, this slide, and then this slide and this slide. That's it. 6 slides, and that's all you'll really need if you're really short on time, by the way, for those of you who plan on cramming this. If you memorize this slide, this slide, this slide, this slide, this slide, and this slide, you're ready for the exam. So yeah, that's it. Now, personally, I like feedback directly, but this is from the PAL team. So Allah yarda alaykum, please fill this form out. And if you have any questions, my details are in the front. Feel free to reach out to me, WhatsApp, email, whatever you guys prefer. Um, or حتى if you see me, stop me, عادي, ask anything. I'm more than happy to help you. And if you have any feedback, if there's things you didn't like in this lecture, if you things you did like, things you'd rather change, I'm more than happy. I'm more than open to, receptive to your feedback. Allah يعطيكم العافية ووفقكم إن شاء Allah. I hope this exam is very easy. Can I review diseases again? Sure, no worries. So yeah, right here. I don't know why I left. Okay, anyways, let me just put this thing right here. Why is this being annoying? My computer's being annoying today. Okay, here are the diseases. You have ICEL, Zellweger, Menkes, and cystic fibrosis, and each one's an example of something. ICEL, basically an acylglucosamine 1-PT deficiency. So it affects lysosomes. How? Because we can't make M6P, so they won't get their proteins. Because we said N-acylglucosamine is what's going to add the phosphate to mannose to make M6P, and M6P binds proteins in the Golgi in order to allow them to go to the lysosomes. So this is a disease where that doesn't happen. Zellweger, mutation in PEX. Basically, the proteins will not enter the peroxisome. Menkes is just an issue with collagen, and we talked about it as an example of a disorder of post-translational modification. In this case, it's cross-linking, but we studied this, glycosylation. And the last one is cystic fibrosis. Basically, it's a disease where misfolded proteins do not get cut. That's it in terms of that. So I hope that's more clear. Of course. Now, if you guys have any questions, let me know. I'm here until you all leave or I get kicked out. This recording stops. But yeah,